Hello everyone and welcome to this webinar. My name is Teresa Riccioni. I'm a Global Pharma GMP Advisor at Particle Measuring System, and today I would like to share with you some considerations about new aspects of the recently released draft of the new European GMP Annex 1. In particular, my presentation will focus on the application of a risk assessment to set appropriate microbiological plate incubation conditions. First of all, I'm going to talk about the requirements, as specifically reported in the Annex 1 draft, of using a risk assessment to identify microbiological plate incubation conditions. And then I will try to provide an interpretation on how the risk assessment methodology can be applied to this particular topic and how to use it in different stages of the manufacturing process. The importance of the quality risk management as a key concept in the new Annex 1 is repeatedly stated throughout the document. In the first lines of Section 1, it is clearly reported that the Annex 1 is a guidance for the manufacture of sterile products using the principle of quality risk management, and that quality risk management applies to the entire document and will not be referred to in specific paragraphs. This means that an important shift in mindset is required, and that quality risk management should be considered a keyword for interpreting and apply the new Annex 1. And above all, that regulatory authorities will expect pharmaceutical companies to perform a relevant risk assessment for each area. And in general, that companies must demonstrate to have a quality assurance system able to fulfill the quality objectives associated with various areas of interest and to have a strong scientific knowledge. In particular, in Section 9 relative to environmental monitoring, it is reported that risk assessment should be performed in order to establish a comprehensive environmental monitoring program, including sampling locations, frequency of monitoring, monitoring method used in incubation conditions. These risk assessments should be conducted based on detailed knowledge of the process, final product, the facility, the operation involved, and historical monitoring data obtained during qualification, and knowledge of typical microbial flora isolated from the environment. Regarding the incubation conditions, the output of this statement is a risk assessment justifying the incubation condition and the culture media to be used to identify all potential microorganisms associated with an environment. In other words, companies should demonstrate that the chosen methods have considered all the risks for maximum recovery of microorganisms. This is particularly important in grade A and B, where the counts are generally very low, and the environmental conditions do not favor microbiological detections. In other terms, the absence of microorganisms associated with grade A environment and the very low colonies allowed in grade B should be the result of a system under control, not of a poor quality of microbiological methods. Let's see how these principles can be translated into practice. To establish the most appropriate incubation conditions to identify all potential microorganisms associated with your environment, start to take information from the USP and the European Pharmacopoeia to have a complete knowledge of the different methods that can be applied. You can also take information from the scientific literature and from suppliers' technical reports to learn about the new methodologies and how to solve technical problems. Then start to perform the risk assessment, considering not only the environment, but also the entire process and the product you are dealing with. And then review data regularly to confirm the effectiveness of the identified conditions. As I said, to perform a risk assessment, you must have an holistic approach that considers the characteristics of your process, including the flow of personnel and materials, the cleaning and disinfection procedures, the characteristic of your product, for example, if it is liquid or lyophilized, the characteristic of your facilities, such as wrong classifications, area difficult to clean, presence of water source in the room, and environmental conditions, such as temperature and humidity, since some of these conditions can favor the growth of anaerobic microorganisms, slow-growing microorganisms, and yeast and molds. Performing the risk assessment to set incubation conditions, you must take into consideration if it applies to an existing or a new department. In the case of an existing department, you must evaluate your historical data, perform a gap analysis to understand if your current incubation conditions are adequate, considering the characteristics of your process and environment. If necessary, perform studies with selected raw media for a better characterization of your microflora. Establish a plan to monitor the development of new microorganisms in your environment, for example by regularly performing characterization studies independently on the monitoring results. And at last, establish a risk review plan. If the risk assessment applies to a new department, you must define the incubation conditions based on a scientific approach that considers the characteristics of your process and environment. Perform initial studies with selected grow media to characterize the microflora associated in your environment. And as for existing department, you must establish a plan to monitor the development of new microorganisms and a risk review plan. How can Contamination Control Advisory Team help you? We can conduct sterility assurance audits to prepare pharmaceutical companies for the inspection of the regulatory agencies or to identify areas that need to be improved. We can make audits to microbiological laboratories or make overall mock inspection in production, quality control, and quality assurance departments. Particle measuring system also provides devices and microbiological plates for microbial monitoring. The BioCap single use is an example of our solutions to make microbial monitoring easier and reliable. The BioCap single use is a disposable microbial impactor to be used with portable or remote microbial active air samples. It combines a media plate and a sampling head into a single unit, eliminating the risk of handling and, as a consequence, the probability of false positive results and final product contaminations, and also the cost and complications related to autoclaving and disinfect stainless steel heads. We have reached the end of this short webinar. Thank you for joining me today. And if you need more information, please contact us or visit our website.